Hi everyone. Now we are going to discuss about a topic called methods of measurement of microbial growth. Especially we will discuss about the bacterial growth. For various purposes, it is often required for the routine determination of microbial growth through the microbial population, microbial cell numbers, or microbial biomass in order to monitor their progress or its progress. Then how can we define a growth? A growth can be defined as an orderly increase in cell components or cell numbers. Coming to the concern of microbial growth, it involves an increase in the number of the cells rather than in the size of individual cells. For example, if we are going to consider the bacteria, the bacterial growth can be measured in terms of two different parameters. The first one is changes in cell mass and the second one is changes in cell numbers. That means increase in the cellular constituents may result in number one, increase in the cell number when the microorganisms are going to be reproducing by budding or by binary fission. So that's how the number will increase. The second is increase in the cell size, where after the nuclear division, they are not accompanied by cell division. So that's how they are going to retain as a one cell only. So obviously the cell size increases but not the number. So microbiologists usually study the population growth rather than the growth of a individual cells. That means they are going to study the population growth than individual cells. There are going to be of many ways to measure this microbial growth or bacterial growth to determine both the growth rates as well as the generation times. And the most commonly employed technique for growth measurements are number one, measurement of cell numbers, where we are going to count the number of the cells both by the direct method, that means direct cell count, and the second one is the viable cell count. And the second one is going to be the measurement of cell mass, where you are going to find out the biomass of the total cells. Then the third one is the measurement of cell activity. That means you are going to relate the whatever the growth to the biochemical activities to the size of the population. So coming to the first one, measurement of cell numbers. So here you can see we are going to have the two types. One is going to be the direct cell count. That means both we are going to count the dead cells and the viable cells. So by the different techniques called as counting chamber technique, then culture counting where electronic counter methods are there, and the third one is going to be the breed method. So that's how we are going to have the three methods in the direct cell counts or total count methods. Then coming to the second type is the viable count. It is an indirect method where we will count the numbers only the viable cells. That means the bacteria which are survived, those only we are going to count. So this is obviously called as viable cell counts. So coming to the first one, that is measurement of cell numbers by the direct cell count method, where we are having the three methods. Those three methods we are going to discuss in detail. Okay, so here the counting chamber technique is the first one, then culture counting is the second one, and the third one is the breed method or direct microscopic method. So coming to the first one, counting chamber technique. So here you can see that in this whatever the direct microscopic counting or count, a counting chamber is going to be consisting of a ruled slide and a cover slip. So here you can see a cover slip was going to be presented or layered on this chamber. So this is an example of the new bar chamber where you are going to have the placement of the sample and then you are going to close that with the covered slip. So here you can see. So this is the counter chamber. Whatever the overflow of the sample is there, there is going to be collected in this grid, and this is a counting grid. And when you observe under the microscope, so this is how the bacterial cells are going to be seen and start off counting the number of the bacterial cells. That means this counting chamber is constructed in such a manner that the cover slide, this is a cover slip, this is a slide, and the ruled lines determine a known volume. It is going to determine the known volume. That means these new chambers or new bar chambers or Petrohofer chambers. So if it is going to be of related to the large eukaryotic, then we are going to have the new bar chamber, or otherwise if it is a prokaryotic microorganism, we are going to consider the Petrohofer counting chamber. So these Petrohofer counting chambers are specially designed slides that have the chambers of known depth and with an edged grid. So here you can see grid on the chamber bottom, and each square, so whatever the each square on the grid has a definite depth and as well as the volume. So in this Petrohofer chamber, we are going to have the placement of the bacterial suspension, or the volume of bacterial suspension is around 0.01 ml and is going to be placed on a microscopic slide with a special grid. Now the cells are going to be counted and multiplied by a factor to obtain the concentration. That means the number of bacteria in a small known volume is directly counted microscopically, and the number of bacteria in larger original samples is determined by extrapolation. So that's how you are going to do this. And the formula used for the direct microscopic count is number of bacteria per cc, that is cubic centimeter, is equal to, you are going to have the average number of the bacteria per large double line square into the dilution factor of the square, large square, so total diameter of the large square, which is going to be of 12,50,000, into the dilution factor of any dilution made prior to placing the sample in the counting chamber. So altogether, you are going to get the thing. For example, if you are going to mix the bacteria with a dye, so that becomes the dilution factor of any dilution made prior to placing the sample. That means in the Petrohofer counting chamber, this is going to possess the small etched squares, which is going to be of 1/20 of millimeter and 1/50 of millimeter deep, and the volume of one small square, so here you can see small square, is therefore going to be of 1/20,000 of cubic mm, or it can be of what we call as 200 lakhs. That means you are going to multiply this one, which is going to be of cubic centimeter. Okay, just remember it is going to be of 1/20,000 cubic millimeter. And how many small squares are there in this large double line square? Means you are going to have how many? 16 small squares are there in the large double line squares that are actually counted, and making the volume of a large double line square is going to be of 12 lakhs. That means 1/12,50,000 cc. Now the normal procedure is to count the number of bacteria in 5 large double line squares and divided by 5 to get the average number of the bacteria per large square. That means you are going to count like this of 5 large double lines, and that is going to be divided by 5. So we are going to divide it by 5 to get the average number of bacteria per large square, and this number is then multiplied by 1, that means 12,50,000, since the largest whatever the square holds a volume of 1/12,50,000 cc. So here you are going to have the cc. To find the total number of organisms per cc in the original sample, the formula that we are using is this one. That means the number of the bacteria per cc is equal to the number, that means average number of the bacteria per large double line square, and the dilution factor of large square, where whatever the dilution that we are taking, and if we are going to dilute it before the sample in the counting chamber, for example adding of the dye, then that factor is also going to be multiplied. So altogether you are going to get the number of the bacteria per cubic centimeter. So coming to the advantages of this counting chamber technique, so this is going to be a very direct microscopic method, very easy, inexpensive, and it is also very quick to count the microbial cell number. And by this method, we can also get the information related to the size and the morphology. And there is no need to incubate the bacterial cell with the help of this counting chamber. And this is mainly useful for counting both the eukaryotes by the humocytometer and the prokaryotes by the Petrov-Hauser count. And more easily, we can count the number of the bacterial cells if they are going to be stained, or if we are going to use a phase contrast or fluorescent microscopic is employed. So these are all going to be the advantages of our direct microscopic counting chamber. Then coming to the disadvantages, we are going to count both the viable and the non-viable. So we cannot differentiate whether they are going to be viable or non-viable. So in this method, dead cells are not going to be distinguished from living cells, and our very small cells are also going to be usually mixed. That means we are going to miss of counting these very small cells usually. And motile bacteria are difficult to count, and this is going to require a high concentration of the bacteria, that is of about 10 million per ml. So if you are going to have this much of the concentration, then it is very easy to count the number of the cells by this counting chamber method. So this is the first method of what we call as a method of measurement of bacterial growth by the direct cell counting chamber method. Then moving to the second, electronic Coulter counter. This is the second method where we are going to use for measurement of cell numbers. The Coulter counter or electronic Coulter counter is an electronic instrument used to count the number of microbes in a suspension or for the direct enumeration of the cells in a given suspension. This instrument, that is electronic Coulter counter, is the instrument uses the principle for particle counting, also known as electrical sensing zone method. In this method, that means electrical sensing zone method, microbial suspension is passed or forced through the small hole called as orifice. On both the sides of the orifice, two electrodes are going to be there. So this is how you are going to have. So two electrodes are there, okay, and here where these two electrodes are present, and these are sensing methods works by using these two electrodes. That means one electrode in the tube with a specific aperture size. So here you can see it is going to a specific aperture size, and the other side, this is going to be the electrode outside the tube. So this is not going to be present inside the tube, it is going to be the outside the tube and immersed in a beaker containing the particles in the low concentration electrolyte solution. Now, an electrical current path is going to be created by the electrolyte solution when an electric field is applied, and the impedance, that means whatever the impedance between the two electrodes, is going to be measured. And this creates the sensing zone. And as the microorganism passes through this aperture, so whatever the aperture is, so here you can see the aperture, they cause a short-term change in the impedance, and this is going to be measured as a voltage. Okay, so that's all happening here. They are going to be of passing through this orifice, and while passing, it is going to measure it. So as this change of the voltage plus and the pulse height is going to be proportional to the volume of the electrolyte solution displayed by the particle. Now the cells are going to be counted, and if a controlled volume of liquid passes through this aperture, obviously particle concentration can be calculated. In comparison to the manual counting, the use of the Coulter principle is advantageous in terms of its speed, accuracy, as well as the precision. So this is what we are going to have. Once again, I'm telling, so this is a Coulter counter electronic method where an electric current path is created by the electrolyte solution when an electric field is applied. And the impedance, that means you can see these are the two electrodes that we are using. So where both sides of the thing you are going to have the two electrodes, okay, and these two electrodes are present, and this is the sensing method works by using two electrodes. I told you one electrode is going to be in a tube with a specific aperture, so here is the aperture, and the other one is going to be outside of the tube and it's going to be immersed in a beaker containing some sort of low concentration electrolyte solution. Now when we are going to supply the current, now an electric current is going to be passed and created by the electrolyte solution when an electric field is going to be applied. So when you switch on the thing, obviously the electric power is going to be supplied and that creates the electric field. And the impedance between, that means impedance that is occurring between the two electrodes is measured, and this creates the sensing zone. As the microorganism passes through this orifice, whatever the thing, from here to through this aperture, this change is measured as a voltage plus. So we are going to have the voltage, and thus the pulse height is going to be proportional to the volume of the solution displayed by the particle. And now the cells are going to be counted, and if a control volume of liquid is going to be passed through this one, so control volume of the thing is going to be passed through the aperture, particle concentration can be calculated. So nowadays we are going to have the automatic machines. That means especially this counter technique where you are going to have the automatic, because of having its high speed as well as accuracy and precision. So this is all about the electron counter, which is the second type in the direct cell counter of measurement of cell numbers. And coming to the third advantages of this counter method, we are going to have so many. I told you that within few seconds we are going to count the number of cells, that is especially in thousands we are going to calculate. And this counter method is especially used or preferable for eukaryotic organisms like protozoa, microalgae, and yeast. And this method is very easy and quick to use, and mainly useful for counting cells, RBCs as well as the WBC2, that is red blood corpuscles as well as the white blood corpuscles can also be counted by using this counter electronic method. Then coming to the disadvantages, this counter is a expensive instrument and not useful for counting bacteria because of their small size, and sometimes this orifice may get coagulated, and non-viable cells are also going to be counted. Even if the dust particles are present, for example, they are also going to be counted. And suspension must be free from any foreign particles, and we cannot distinguish living from the dead cells here also. In all the types of direct method, we cannot distinguish the live cells from the dead cells. So we are going to count the total number of the cells. That's why we call it as total counts or direct cell count methods. Now moving to the third method of direct cell count in the measurement of cell numbers, that is the breed method. So here in the breed method, a known volume of cell suspension of about 0.01 ml is spread uniformly over a glass slide with a specific area of about 1 square centimeter. So here, for example, you can take a slide, the plain glass slide, so where you have to measure, and that area is going to be of a specific area. It is going to be of 1 square centimeter area. And now in this area of the smear is going to be fixed. That means you are going to take the bacterial suspension and you are placing and you are spreading it, and then you are going to fix it by the heat fixation. So that means in this area only the sample is going to be fixed. It is not going to be of overflowing this side or that side. So this is how you are going to have your whatever the sample or suspension you are taking. Then this suspension is going to be fixed by heat fixation, like how we done in the gram staining or staining purposes, and then it was going to be stained and then examined under the oil immersion. Okay, so where you are going to observe the cells very clearly, and then the cells are going to be counted. This counting of total number of cells is going to be determined by calculating the total number of microscopic fields per 1 square centimeter area of the smear. So that means you are going to take this 1 square centimeter area and you are going to find the total number of microscopic field. So microscopic field means the field at once you are looking into the microscope, whatever you are observing, that is going to be called as field. So you are going to take a number of microscopic fields that is going to be multiplied by using the area of the smear. So altogether we will get the number of the cells per 1 square centimeter. So let's see how we are going to calculate it by the calculations, mainly having the area of microscopic field. So we know that in any circle we are going to have the radius, so that is going to be of r, because when you look into the microscope the field is going to be of circle in manner, so that radius is going to be calculated first, that is pi r square, and the oil immersion radius is going to be of 0.08 mm. So that how the area of the oil immersion is going to be of pi r square into 3.14 into 0.08 mm square. So that is giving rise to 0.02 square millimeters. So remember, this area that we have calculated is going to be the millimeter, and we know the square that is area of the smear is going to be in the centimeter. So obviously we have to convert that into the millimeter. So that's how 1 square centimeter is equal to 100 square millimeter. The coming to the number of microscopic fields, that means how many times you have moved the slide and looked into the microscope, those are all going to be considered as number of microscopic fields. For example, if you are going to consider then 100, that means whatever the area of the smear is there divided by area of the, what it is going to be the area of the microscopic field. So you have to divide these two, and altogether the number of the microscopic fields will be 5000. And now if we want to have the number of cells per 1 square centimeter or per 0.01 ml, because we have taken that much of the sample only, isn't it? So that is what, either 1 square centimeter or you are going to calculate the number of the cells per 0.01 ml. So now that is going to be getting by multiplying the average number of the bacteria per microscopic field. For example, if you are going to take the count of 100 average, you are going to take the, for example, here you had the, at the first field you got 50, in the second field you got 2. You have to add all the number of the bacterial cells in the different field views, and then you have to take the average of it. That means if you move the slide for 10 times, and then if you are going to have the number of the cells, for example here 50 plus 20 plus 10, like this, like 10 times if you are going to take altogether. So for example here is 3, so I have given here 3, no? So 50 plus 20 70, 70 plus 10 80. So 80 by 3 is going to be the average of it. So obviously you are going to get the average number of the bacterial cell per microscopic field, that have to be divided, multiplied with 5000. What is this 5000? It is the number of microscopic fields. So once you are going to multiply this value, that is average number of the bacterial cell per microscopic field, that with this number of microscopic field you will get the number of cells per 1 square centimeter. And the way through which you are supposed to move the slide is going to be in the zigzag manner. So here the arrow is going to show. That's why I have kept this picture to know the way in which you are supposed to move the slide. Okay, so there will be no lines. So this is related to the Petrohouse or chamber counting. There also you are doing the same thing. So this is what about the breed method by which you are going to calculate the number of the cells. Then coming to the advantages, here it is going to be a very quick, obviously, and easy and inexpensive when compared to the electron counting chamber. And then we can calculate because we know the oil immersion radius, everything, and it is very easy. Then coming to the disadvantages, Here we are going to count both the viable cells as well as the dead cells. So we cannot differentiate the viable cells and the thing. And some people, those who are operating this kind of the method of calculating the number of cells, have to be of very perfect in handling the microscope and counting. There should not be no confusion of reoccurrence of the same field counting. So with all these things, that means the first one is a counting chamber, then culture counter, then breed method. We are going to calculate the or measurement of cell numbers is going to occur by direct cell counts or TEL. That is total count we are going to take in these three methods. What are those three methods? One is going to be the counting chamber technique. The second one is going to be the electron culture counter, and the third one is going to be the breed method. In the next part, we are going to discuss about the indirect methods of measurement of cell numbers. So where we are going to discuss or we are going to count only about the viable counts. Okay, that's all about this part. Thank you.